Okay, so I'm just going to make Hussam presentation a little bit more complicated, I guess. Yeah. So as Hussam talked about all the sequencing, so again, we isolate RNA from those samples and then we sequence them and then we have some reads and then we align those reads to the horse genome. But there are two steps that I want to talk about before I get to my presentation. One is about this isolation of RNA from our sample, and the other one is the alignment to the horse genome. So it is known that majority of the RNA in the eukaryotic cell and also mammalian cell are not mRNA, the one that we are interested in. Actually, the majority are ribosomal RNA and also transport RNA in the cells. And those mRNAs less than 5% of the total RNA within the cells. And this is unrelated to the way that you extract RNA or prepare your library when you are doing the RNA sequencing. Regardless of the way that you are preparing your library, you still get the mRNA, which is the majority of them, but you still also have other RNAs, including those ribosomal RNA in your data set. And those, as Dr. Erd mentioned earlier in the morning, those 16S ribosomal RNA are one of the methods to make the phylogenetic tree to see what are those bacteria and what are the phyla of those bacteria. The other thing about the RNA sequencing is when you align it to the reference genome, in our case, to the horse reference genome. It's not 100% thing. So most of those reads that you get, you align it to the horse genome, but not 100% of them will map. So what are they? Are they junk? So for example, in the Nocardioform data set, around 80% of the reads that we received were mapped to horse genome. And in another data set that we have, the pregnancy data set, around 90% of them mapped to the horse genome. So the question was that, okay, what is that left over? Are they junk? Are they just error, or can we use them? So we came across this study that what they did is that they transfect some human cell, HeLa cell, with a JFP salmonella that express JFP, and then they sort them and they just collect those salmonella and those cells that were infected with salmonella, and then they extract RNA from those and did sequencing. And what they found is that they could isolate ribosomal RNA from salmonella in those RNA sequencing data. So then we thought, okay, can we look at the ribosomal RNA in these sequencing data that we have to see what kind of bacteria we have? So again, the methodology is very similar to what Hussam just presented. The only difference is that this is another set of sequencing and a little bit deeper sequencing, but the rest, we have the lesion sample, we had the normal region of the same placenta, and then also the control samples. So I'm not going to talk about bioinformatics, I'm just jumping to the result. So yes, we were able to look at the 16S ribosomal RNA from the unmapped section of the read. And based on those ribosomal RNA, we could see that two of the sample, they had very high abundance of the actinobacteria in them. So in the majority of the sample, the Proteobacteria was the most abundant bacteria. And almost all the samples, they have certain amount of actinobacteria. Again, back to the talk in the morning, probably they can be contamination, or I still don't know if we can call it a flora or something, but there even in a control sample, there are a certain amount of actinobacteria and Protobacteria. But what was obvious in two of the sample, this lesion 1 and lesion 4, there was very high abundance of these actinobacteria in the sample. In the next step, we look at these bacteria at the genus level by mapping all those unmapped reads to the prokaryotic reference genome to see what genus of the bacteria that we have. And again, I'm sorry, this lesion 1 and lesion 4 were the one that has high actinobacteria. Now we could see that they have a high abundance of the amyclotopsis in them. So again, it matched the report that we had. And these are Nocardioform placentitis, and we had high amyclotopsis. But the other two lesions, the other two sample, lesion 2 and lesion 3, we didn't detect the high abundance. We still detect a certain amount of amyclotopsis, but it was not one of the highly abundant genus of bacteria in these samples. And once we cluster those samples based on their bacterial genus that they have in them, we could see that again, lesion 1 and 4, that they had a high actinobacteria or amyclotopsis, they clustered together, and then another one were separately. So we wanted to see if there is any association between the bacterial genus in these samples and the transcript of the placenta. Again, I'm not going to talk about the transcriptome of the placenta. Hussam just talked about it, but here again, this is the heat map of the gene expression of the placenta. So each line represents one gene, and a red represents high expression and green low expression. And as you can see, again, those lesion 1 and 4, they had very similar gene expression, and then lesion 2 had another pattern of gene expression, which caused it cluster by itself in a distance. And if we put these two together, so if we cluster these samples based on their bacterial genus, so we have lesion 1 and lesion 4, and then we have lesion 2 completely separate based on the bacterial genus. And then when we cluster them based on the transcriptome of the placenta, the gene expression in the chorioallantois, again, they cluster in the same way. So there is an association between their bacterial genus in those samples and the gene expression in the placenta. But then the next question was that, okay, what else we can understand? Now we can see that what kind of bacteria are there based on their ribosomal RNA, but can we look at other things? And yes, we could look at the mRNA at those samples. Again, in this data set, they were able to look at the mRNA in those cells. And this is the base of the technique called dual RNA sequencing, which is when you sequence a cell containing infected cell, so you sequence them and then you separate bacterial transcriptome, then a host transcriptome, and then you can see the association between those. And based on this dual RNA sequencing, they were able, in several studies, they were able to find a specific stimuli that coming from those bacteria, the gene expression from those bacteria that cause that response in the host, and it helped to understand the pathogenesis of the disease. But there is one thing about this technique that is require very deep sequencing. And this is based, again, on the fact that there is even in an infected cell, there is a very minimal amount of bacterial RNA in the cell. And out of that amount of bacterial RNA in the cell, again, majority is ribosomal RNA and transport RNA. And the small portion of that is mRNA that we are interested in. That's why we need very deep sequencing. So for example, at least 200 million read. And the data set that we have, the data set that Hussam present is around 8 million read, and the data set that I'm presenting is around 30 million read. So we need way deeper sequencing to be able to look at the mRNA at the bacterial level. But still, we wanted to look if you can see something out of those. So what I did, I map the unmapped section to the amyclotopsis reference genome, and this is the gene expression of the amyclotopsis from the data set that we have. And again, you can see those lesion 1 and 4, they had one pattern of gene expression of amyclotopsis, and then lesion 2 had another pattern. And when you look at the function of those genes, these lesion 2 They were all showing that they are metabolically active, they are biosynthesized other metabolites, while these genes from lesion 1 and 4, they were not showing any active pathway, but there were few of them that were similar to the quorum sensing genes that were identified in Klebsiella. And again, just to remind you, these lesion 1 and lesion 4 were the one that had very high abundance of amyclotopsis. So there is a chance that, again, it's just hypothesis, that these bacteria, they reach to a certain level and they are not metabolically active anymore, while the lesion 2, that it had a low abundance of the amyclotopsis, it start to be metabolically active and start to grow to reach to a certain threshold. And again, this gene expression in amyclotopsis within those sample, again, follow the same pattern as a gene expression in the placenta. And the other thing that we did is that we look at the gene expression of the Klebsiella in those sample. And again, only one of them, that lesion 2, that was clustering separately from the other lesion samples, were the only one that has a high expression of the certain genes from the Klebsiella in the sample. And again, those genes were associated again with the metabolic process and responses to stimuli. So with that, we're just proposing that we need to repeat these samples in a bigger sample set with a higher sequencing depth to be able to look at the association and then some correlation between the gene expression in the bacteria, and then translate that to the placenta. Thank you.
Thanks, Puya, for a nice job sort of looking at the potential of these kinds of approaches. We've had a couple of discussions now about high throughput sequencing and its application and looking at some of these diseases. Are there comments or questions from anyone?
Just as a suggestion, I thought, that will be interesting to look at the microbiome of placenta, probably from the healthy foals, with or maybe without or maybe with placentitis. The samples taken from the surgery, the obstetric, surgery, and see, if the placenta is really sterile when you take the specimen, after that surgery, obstetric surgery.
So it's really, I mean, there are lots of debate around it, if there is a flora in a placenta or not, even in human.
C-section surgery I'm talking about, C-section surgery.
Yes, even with the C-section. So they did in human, and there are some paper that they saw some bacteria, some flora of the placenta, and there are other that there is just, the paper came this year that from in Nature that they say, no, it's all methodologic error. If you see bacteria in the placenta, they still think that it is sterile. But then here I just got involved in a study in monkeys, and we see bacteria in the monkeys placenta. So yes, it's interesting, and it's hard, and it needs lots of control to prove that it's not a methodologic error, but it's very interesting to know if there is something over there or not.
Anyone else? Okay, thanks, Puya.